Yeah, we can see your screen.
Okay, we make progress, didn't we? Very good. Thank you all for joining in my presentation here. What I decided to talk about is actually a problem that we've been addressing for the last 3-4 years. And what it is to put an array of sensors. It's basically a protein array that we spot on a piece of paper, nitrocellulose. And we put that on a centrifugal platform, basically a spinning compact disc. And we do that for bacterial identification and quantification. So we call that ID/Q. And antibiotics susceptibility testing, AST. So what it means, it's a long title, I know, a lot of words there, but what it means is this. If you have a bacterial infection, we identify what the bacterium is, and then we test what is the best antibiotics to get rid of that bacterium. And so that is the susceptibility testing. And we are trying to adapt this platform to what we call extreme point of care, or EPOC, meaning it's not your regular nice central laboratory setting in a hospital. It might well be in a remote place where it's not so easy to have access to highly specialized clinicians or so. So what I will cover is what is extreme point of care. So explaining a little bit of the circumstance where these things are made. Then why EPOC on a CD? So why do we think putting an extreme point of care test on a CD, both in terms of the hardware and the disposable? The hardware will be the box where you put your CD in, and the CD is the disposable. And to kind of make you familiar with the operation of that instrument, we will give some example functions. And then we come to the major part of the discussion is how can we do microbe identification and AST, so antibiotic sensitivity, susceptibility testing in less than 2 hours. The image I show on the right are example products in the Marc today, but doing rather simple things. On the top, we see at the top right is a glucose sensor. Then on the top left is Abbott's iStat device that next to the patient gives you information about the sodium level, potassium, pH, oxygen, CO2 in your blood. And the Gene Expert at the bottom is an instrument from Cepheid, which is already a little bit more sophisticated. Here we do PCR or amplification, DNA amplification. And it's that kind of instrument we want, but it needs to do a lot more, right? It needs to do identification of a microbe and then showing what kind of antibiotic should I use if that's the microbe you're infected with. Now, this has become a very, very big problem, this antibiotics testing. Why? Well, in many countries, including in India, you know, this gentleman here basically makes a living by prescribing antibiotics, but it doesn't mean that you need antibiotics or if it's the right antibiotic. And what that has caused is that worldwide, all mankind is now exposed to more and more aggressive microbes, and we don't have the right antibiotics anymore. Most of them have run out of efficiency. So it is a huge problem. But I'll talk more about that when we get to that point. So what is extreme point of care? Well, you know, there's so many challenges. If you look at rural clinics in extreme settings, you know, there's lack of trained personnel, long turnaround times for results. You know, when you do culturing of microbes, you might have to wait 3-4 days, maybe a week to get your results back. You're working in harsh environmental conditions, limited electricity, cost limitations, and lack of a complete test panel. You know, you would like to be able to test for as many possible pathogens as possible, not just one, because you happen to be in that environment. It's difficult to get there, so you would like to get as much answers as possible from one device. So arrows below these identified challenges do tell about what we are trying to do with our system. By the way, here you see the disposable, right? This will be our compact disc, and this is actually has the design for the very test we are talking about today. And as we go along, I hope I will be able to explain how these channels and chambers in this plastic disposable will allow you to read the identity of the microbes you identify, and also let the doctor know what antibiotic to use. So, but getting back to challenges and how to tackle them. So if we could have a complete lab in one system and automate the processing for diagnostics with built-in detection readout, we would make the life of people doing these tests in the field much simpler. In other words, you kind of own a box with just one button. You push the button and that's all you need to do. You do not need to have a high-level education to run the machine. Harsh environmental condition, that is one of the big challenges. And for this group here of engineers, you know, I urge you all to look at how can we, for example, maintain a small amount of liquid in a pouch of some kind in an environment where it's very hot, where there's high humidity, and that chemical stays intact. It is a very difficult challenge. You know, mind you, we have to store some chemicals on this platform, and how you do that. I'm mentioning here biomimetic environmental control because lately we have started looking at how in nature life that always contains, you know, some liquids, how does it protect itself from drying out? Think about spores that can survive very, very harsh conditions for a long time. Limited electricity, so we are talking about wireless, low power implementation and power harvesting. You know, maybe just you carrying that little instrument generates some movement that can help power the device. Cost limitations, the motor running this, we want to be very small, simplified, and in a way, you know, we want a complete lab on this disk. That's why we call this lab on a disk. And we want to have critical test panels. For example, would be a fever panel. What that means is there might be 20 or 30 reasons why a patient comes in and says, I have a fever. You know, it just could be a cold, but it also could be related to HIV. So a fever panel would allow to screen where does this patient need to go, home, or perhaps be helicoptered out, let's say from Karapur to Kolkata, because it's that urgent. By the way, I have a review paper that we wrote on CD-based microfluidics for primary care and extreme point of care settings. It's quite old by now, 2016, but if you have an interest in this, I urge you to check that reference out. It was worked on with colleagues in India and in South Africa. The picture on the bottom here kind of shows this concept of what the spinning disk needs to replace. You know, it needs to replace a PCR thermocycle, sample concentrator, a centrifuge, an incubator. It needs to be coupled with your cell phone. It needs to have a cell lyser. It needs to act as a microscope, and it needs to be a mixer. So that is in a way our big challenge, to put all of that in one simple instrument. By the way, I'm showing again some pictures from India. These, I believe, I'm not 100% sure, but I think this is a picture that we took together with Satad Al-Saha, who's a dear colleague who's running a kiosk system in West Bengal. And I believe that is also from another kiosk place. These are the places where these kind of machines would be run. So now I told you a little bit about the extreme point of care. Now we are to the point where we're going to talk about the hardware and then the disposable for this extreme point of care. So as I already told you, we are using a CD, but we are also looking as much as we can to the inside of a CD player to see what else we can use from the machine. And a CD player is actually, obviously there's very few of them left in use today, but it was quite sophisticated for very little money. You know, you could buy a Discman playing your music through your earphones for $25. And look, it had a linear motor, tracking motor. It had a regular rotating motor. This allowed you to go to any place on this disc, you know, and in the case of music, that was perhaps to start at the beginning of a certain song you like. In our case, we're going to use this as an XY platform. In other words, let's say I know that I have a protein immobilized right here on this XY coordinate. And I can go find that spot. And then if that spot lights up, perhaps it means you have that bacterium DNA, that microbe DNA in your blood or saliva. Right, so here you see the same. This is the disk drive, the tracking drive, linear, and then the rotating drive. And here we have the laser pickup assembly and laser lens. What that does is basically the following. As you're spinning your CD, a laser is reflected off the spinning platform. There's a layer of aluminum metal, and small bits have been injection molded into your CD. And when the laser hits one of these pits, you can imagine the light reflected is different than when there's no pit. What that does on the optical pickup, you either have high intensity or low intensity. And that's translated into a digital signal. And that gives us basically zeros and ones, right? But you could convert that from digital to analog by this DAC, digital analog converter. You get your analog signal, perhaps sound, data, whatever it is. Well, I shouldn't talk about data here, I'm talking about analog. But for example, you get your sound out of that system. So you kind of recognize that, yeah, that comes from the music industry, film industry, but you start also recognizing all of these things you also use in clinical settings. For example, I already told you, this is an XY stage. If you have an arrayer for protein array fabrication, that's basically what you need, an XY stage, and you have that in that system. Since it's a spinning platform, it's also a centrifuge for centrifugation, but in this case, it's a smart centrifuge, right? It can be stopped at any time, it can go to different places, so it's better than your big centrifuge. Of course, it being smaller, you might have to spin faster to achieve the same G-forces. Then interestingly enough, this is also a microscope. It's a microscope because if you don't digitize this signal here, and instead of you're looking at these pits, let's say you put the wing of a fly or a bug on this platform, you can actually have a nice, very high resolution image. And in this case, actually, we are imaging an immunoassay where each of these dots is a protein spot. And you can also visualize it just like you visualize this wing here. This shows the microscopy with it. So it's also a pump, of course. If I have liquid here, I can pump the liquid towards the rim of the disc. It's an inexpensive vortexer. Vortex is like a mixer. That's kind of logical, right? If you spin this and while spinning, you have a moving membrane, sorry, a moving metal disc here. I'll explain that. I'll come back to that later. That metal disc can destroy the cell walls and release the DNA, the proteins that are inside the cells. And that's what you do in a vortexer. And so, but don't worry if you don't understand that one yet. I'll come back to it later. And then it's also, of course, an inexpensive disposable plastic disc. So this disc ought to replace, you know, these tubes you're used to use, these microscope slides, etc. Right? So that's why this hardware is, in our opinion, of big interest. And look to kind of make that even more clear to the mind. What we have done here is we have taken all of the equipment that you have in the lab, typically, we put the cost of this, right? And if you compare the total of all of these instruments, you end up with, just a second. I'm on the Zoom link. I'm on the Zoom. Okay, sorry for that. So you can see that that equipment would add up to about $170,000. And now we've been able to replace all of this in this piece of equipment that ends up costing about $200. And my students can put a system that like this together now in about 1 week. So that's kind of the message here, that you could bring all of that to much more humble places than your typical central lab in a big hospital. So now I told you about the instrument. What about the disposable? So we already know the disposable is a disc, as you can see here, plastic, injection molded. And well, that disposable actually has a lot of advantages. So for one, it's easy to multiplex with CD fluidics. What that means is I can do many tests on the same platform. You know, you see all of these similar elements. These are repeats of the same test. In other words, I multiplex a whole bunch of analyses on that platform. I also don't need any tubing to put things together. You know, the tubing is embedded in these plastic channels. Now, from the physics point of view and the operational point of view, this spinning allows you to access three major forces. You know, one is centripetal, so that means I can spin liquid from the center outwards, right? Centripetal force will go this way, outward. I can also use the Coriolis force, that is depending on what direction I'm spinning in. I might be able to push liquid, let's say, in this chamber or in that chamber. So that's to go to the left or the right. And then finally, the Euler force, that's when the RPM suddenly changes, that will allow you to mix liquids, because now the liquid suddenly gets this force to push forward and perhaps burst into another chamber or mix with another liquid, maybe a dye or another reagent. Then also, this platform, it doesn't care what the pH is, the ionic strength. And also, you know, you could easily put in milk, blood, urine. It's quite forgiving in the nature of the sample you're using. It's an inexpensive disposable plastic disc. No problems with bubble formation. Very, very often a problem in fluidics. You know, if a bubble sits on top of your optics, your analysis is over, it's done. But here, because of the spinning, you're spinning out the air. Just because of the motion, the liquid will go to the right, and the air bubble will go to the center. So you're getting rid of bubbles. It's a closed self-venting system. That means you can keep it all self-contained, so there's no disposable problem, no disposal issues preventing contamination. It's easy to perform mixing, volume definition. So pretty much anything you imagine in terms of analytical processes that are going on in a typical laboratory, you can do on this platform. And over the, you know, about 22 years ago when we invented this, pretty much all of these functions have been demonstrated. And then importantly also, the fluidic network, you know, all of this is not coming in contact with electronics or the pump or the motor. The pump is actually CD, right? But the motor that spins this, you don't want liquid to go on there, but that's nicely isolated. And by the way, so in all these years, we've been doing research on this, products have come about. You know, here is the ABAXIS system where blood panels are taken from, in this case, animals. This is for veterinary application. Imtech, also blood panels for humans, and Samsung, an ELISA assay. There's many more new products. Actually, from all of those, Samsung has been bought up by company Nexus. That's now in San Diego, who we work with. Imtech, this is more research, and ABAXIS is actually the oldest, most established centrifugal fluidics-based commercial company. So, okay, what have we done so far? We talked about the instrument, we talked about the disposable, we talked about extreme point of care. And as I told you before, talking about this major application we're gonna run today, I want to tell you first how this whole thing operates, how this CD machine works. And I will illustrate it first by illustrating cell lysis. And so please follow with me before we show this little movie here. I have my plastic disc, and embedded in the disc, so these cavities here are closed, right? There's a plastic disc on top and bottom, and the design is retaken here in this drawing. And what it shows is chamber 1, chamber 2, and this is a metal disc that's locked into this top chamber in this one here. And you can see the metal disc also here. So this is all the same chamber, this top one. And then when you see the blue, that is an opening to air. So that is where you put your sample in. You pipette it in here. And then, so that is also blue, that's also blue, that's also blue. This is a capillary opening between the two chambers. And then this here is a siphon, this elbow, with the crest of the siphon being on top here. So what we're gonna do is this unit is put on top of a platform. So these are all of these units, and that's a magnetic holder with magnets. So you see these magnets here distributed in a certain way. And what these magnets are doing is when this is rotating over the magnets, they are attracted to the permanent magnet underneath. So this disc that's movable up and down, so this disc can move like this, it's attracted at this point to this permanent magnet underneath the platform, right? Let's say this one. And now as this moves, this guy will be attracted to this permanent magnet. That means the disc will go up and down. And so now you can imagine, you see this happening, juk, 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 juk. This is being pushed up and down because of these magnets underneath this plastic. Now, I also have cells in here, plus I also add glass beads. And I do that so that when this magnet goes up and down, the cells are really broken up. And that means I have now cell wall debris, I have DNA, I have protein, I have RNA, organelle fractions that are all accumulated in this top cell. Now, this step that I'm describing here is rather slow because you couldn't go too fast in RPM, right? Because this heavy disk wouldn't follow anymore. So after the lysis is done, I now go to much higher RPM. This doesn't move anymore, but the liquid in this chamber is able to go to the bottom chamber because the centripetal force pushing on the liquid can push it to this capillary where surface tension is big, but surface tension is overcome by the gravitational force pushing the liquid from this chamber to the bottom. So now I have this liquid with all of these fragments at the bottom, and I happily spin this for a while. Centrifugation happens so that the heavier elements settle at the bottom, and the proteins and the DNA, the smaller fragments that you want, accumulate on top here. So at a certain point, when I think I have enough fragments that I need, when this is high enough, I slow down the spinning, and now this top material that's above this intersection here can move up against the centripetal force, because now surface tension is dominant, and empty in this chamber. So let's go over this in the video. Lysis already done. Now the liquid goes to the bottom chamber at a high RPM. See, the liquid has gone above this T. Now I slow down so that the liquid can rise up. You see it go up. And voila, it has gone over the crest of the siphon, and it's ready now to do something else, perhaps PCR, but we have neatly clarified, purified, and isolated DNA and proteins, etc. So that is one function to kind of teach you on how this platform works. So lysis happens at low RPM with oscillating steel member, that disc, and glass beads. Volume definition and clarification is at high RPM, right? Volume definition, everything that above this line will be able to be sucked into by capillary forces all the way over the crest of the siphon. So transfer of clarified liquid with nucleic acids and proteins to the collection chamber via siphon at intermediate RPM. So let me hold it here for a second and just see how my audience is doing and if you're all following. So let me see your faces here. And I'm just opening here a little bit for some questions, because as you know, I need to give one more example and then go really into the discovery of this microbe identification and antibiotics susceptibility testing. But so I'm open for a few questions before I go on. I see we have about 38 people. That's a good group. I'm also seeing if there's people I recognize or remember from Suman's group.
Yeah, guys, you can ask any questions that you have.
So one question.
Please. Go ahead.
What is the RPM range that you get through this device?
Yeah, so we rarely need to go above 6,000 RPM. In your CD player, you'll go about 3,000 RPM. But here, like the functions I was telling you about, you're not going higher than 3,000 RPM, rotations per minute.
Yeah, thank you, sir.
Okay, so now the second application, and this very application is the one we will need, because this indeed involves an immunoassay. So you see this paper here that's been put in what we call a reaction chamber. And this could be any kind of antibody or antigen to carry out an immunoassay. Right? And so, for example, in this case, the antigen is at the solid phase, the paper. And if there's a reaction with involving the primary antibody and the secondary antibody, this enzyme that's linked onto the secondary antibody changes a substrate that's colorless and is in solution towards a product that's dark blue and precipitates. So when the test is done, you would see this darkness, and the darkness would mean, yes, the reaction has happened, and you would pick that up by an absorption measurement in this case. Now, what I want to illustrate to you is a very important aspect of microfluidics. You know, if you have a sensor like this array and you send liquid over it, you know, you go from the center and you go towards the rim. The liquid that's here at the rim, it's gone, right? It's over, it goes to waste, to this waste chamber here. But suppose your test isn't done, you want that liquid back. How do you do that, right? Because if you spin your CD, let's say, you know, I'm spinning this, the liquid goes from here to here, but say my sensor array is here and I need the liquid back. How do I do that? Can I bring the liquid back into contact, and maybe several times still my assay is ready? Well, that is how we do that. Look, say I'm loading this liquid here, right? And I want the liquid to go back up. So first, the liquid is loaded. I'm now starting to spin. The liquid goes down, right? Central peak force goes this way. And I keep on spinning. I bring even more liquid in here, so the RPM increases. And now what happens, this chamber here is closed. And what that does is the liquid compresses the air. And the higher in RPM, the more this liquid is compressed into a smaller and smaller volume. So if I want the liquid to go back, you know what I do? I go lower in RPM, I slow down, and you can see the liquid goes back up. And that means if I put a sensor here, this liquid can go back and forth over the sensor, right? And that's what we've done here. You know, that is a pressure chamber. That's the loading chamber. And we can make the liquid go back and forth till we get the right exposure of the sensor array. This going back and forth is illustrated here. The center of the CD is here. Normally, you know, you bring it down, you can never go back up. But if this is a closed chamber, we can go back and forth as many times as we want. So we do reciprocation of fluid flow by alternation of high and low angular frequencies. And that maximizes the molecular encounter between the liquid phase and the solid phase. And the advantage of this are manifold. So here you have the same paper strip, and you know, we multiplex it, we can put a whole bunch on the same CD. Now, imagine that all I do is put a liquid over the array and I don't move anything. That is what you would get in terms of concentration of the reaction product as a function of time. It would be slow, right? And I wouldn't get a big signal, and we call that passive. Now, if you flow through, of course, everything goes to waste, right? And you need much more liquid, 300 microliter, 30 times more than if the droplet is just sitting over it. But look, I have a much better signal in a short amount of time. But it's 300 microliters. Now, if I reciprocate, I use 10 microliters, much less than here, and I get about the same dose response curve. So this trick allows me to be faster and use 30 times less analyte. So now I think we are weaponed to start discussing the main application that's at hand today. So this is all about making sure that the doctor prescribes the right antibiotic. And for that, you know, we first need to look at how has this happened? Why is antibiotic resistance increasing? Well, imagine you have a lot of germs and a few that are drug resistant. So the antibiotic you add will kill the bacteria causing the illness, but it will also kill the good bacteria, protecting the body from infection. The drug-resistant bacteria are now allowed to grow and take over. So, and some bacteria give the drug resistance to other bacteria, causing yet more problems. So, you know, we all know that global warming is a big crisis, a big problem, but this one is also a really big issue. You know, some people are saying that a failure to address this problem of antibiotic resistance could result in 10 million deaths, unnecessary deaths by 2050, and costing a whole bunch of money. So how can we deal with this? Well, one is you develop new drugs, and the other one is you stop abusing antibiotics, right? Now, the problem about developing new drugs will become very, very clear in a moment. That doesn't seem to be a feasible path. So the path we will be on is stop abusing antibiotics. And so why is developing new drugs not a likely path? Well, here you see over the years, the adaptation of antibiotics, you know, starting with the very famous and, you know, first ones, penicillin. And you can see more and more were discovered very, very fast, right? But then you will see that as time goes on, look, from 1990, a huge discovery void. And then here you see the resistance identified. Look, penicillin 1928, resistance 12 years later, right? And so you can imagine this is scary, right? We cannot obviously fast enough beat the bacteria until winning the war against us. So what do we do? Well, as I said, it doesn't look very promising finding new drugs. So no new class introduced as treatment since 1987. And for gram-negatives, no new classes since 1962. So why is that? I mean, there's many reasons that have contributed to this, right? So in the current paradigm, you know, what happens is patients are treated with best guess empiric therapy. You know, you have a prescription, sample is tested and cultures. There's 3 days that passed here. The treatment is either over-treatment or under-treatment. So there is a problem here with my... So this is how we currently do it. How we want to do it is, instead of the patient having to go home and basically the doctor observing, does this antibiotic I prescribed work or not, we do on-site testing. We figure out this is the bacteria, the microbe you're infected with. So we have an accurate prescription. And we can describe the ideal amount and type of antibiotics that this person needs. That is what we want to do. So now, and I see time is going fast, I will shorten my talk a little bit so you guys will have time to ask me questions. But so what is our assay? What is the solution we have come up with? Well, we are looking at a ribosomal RNA. And specifically, we're going to measure the growth of ribosomal RNA. Why? But first of all, a ribosomal RNA has an excellent metabolic activity. What that means is it responds very rapidly to environmental factors. So suddenly this antibiotic is present, immediately the amount of ribosomal RNA responds. So it is almost like doing the culture that you normally do, which is of course phenotypic, but doing it much faster. Because as you know, culturing takes too long, 2 to 3 days. But by using ribosomal RNA, where you don't need to do the whole length of culturing, by looking at the RNA that very, very quickly, faster than waiting for the cells to grow, responds to its environment, especially metabolics. There's an inherent amplification involved because during incubation, the short time we incubated, the ribosomal RNA very, very, very quickly multiplies. It's almost like, okay, when a cell is ready to divide, the ribosomal RNA are the engines to make the division possible. And so there's a huge exponential increase of ribosomal RNA. Then to make it a good probe, it's very specific. And you can find, this is an example of ribosomal RNA, you will find both universal and species-specific sequences that are accessible to hybridization by probe pairs. So that is our trick. Now, how does that work in more detail yet? So I'm looking here at the ribosomal RNA number of copies as a function of time. In this case, this is the growth curve when there is no antibiotics present. The next one is the curve when you have resistant bacteria. You know, they act as if there's no antibiotics. But then when there's bacteria that are susceptible, those, the ribosomal RNA copies, decrease dramatically. So What is the measurement? The measurement will be after 90 minutes, which is an hour and a half, we have a reduced percentage of ribosomal RNA in susceptible bacteria compared to the positive control. So this is what you measure. You measure this difference. So the ratio is bacteria incubated with antibiotics over the positive control. And basically, we're doing this just showing. So that point there is ratio of resistant bacteria over positive control. That one is susceptible bacteria over positive control. So you do the ratio of the ratio, basically, right? Okay. The assay. I guess this is the most important slide. And then how we translate that to the assay on the CD. Okay, so the top structure is the identification and quantification, which we do in 30 minutes. So let's go over that first. I have my bacteria sample. I do the lysis to release the RNA. And you know the lysis already, right? That was my example. You use that magnet oscillating, magnets that cause that disc to oscillate. And in 6 minutes, we release the ribosomal RNA. Now we have a capture probe to capture the ribosomal RNA. We hybridize with the probe in 15 minutes, and we have a detector probe that attaches to the ribosomal RNA. And if the bonding is successful, we do detection in 5 minutes with fluorescence. That's a fluorescent probe. So identification and quantification in 30 minutes. Now, the antibiotic susceptibility test is remarkable similar. You see, you do all the same, except you do the incubation in the presence of antibiotics. And you can see all the other steps are identical, which is very good, right? Because now you can use the same element on your CD to do the both. This one, what is it? This one, what is the antibiotic that kills it, right? What antibiotic causes the curve to go down to the blue shape? For what bacteria, what microbe? Very good. So how do we implement that on the CD? So this is my incubation chamber. So that is where I introduce my microbes. This is the cell lysis. So you see the same structure we learned about. That's moving up and down. There's one difference here over what we did before. We also need sodium hydroxide. So this is both mechanical lysis and chemical lysis. And because I have that sodium hydroxide, I need to neutralize the liquid that comes from this chamber, because otherwise you cannot hybridize in alkaline medium. So here I neutralize, and voila, I go over my hybridization chamber over here. And after hybridization, I do some washing to get rid of unselectively bonded material, and I do the detection. So assay steps for ID, identification, cell lysis, neutralization, hybridization, with detector probe detection. Okay, AST, all the same, except, of course, we always need to do sample loading, right? Incubation with antibiotics. And so we're putting up to 11 different antibiotics. We also have 2 controls, and we have a universal probe. That means we need 14 chambers in total. And that means we can test a bacterium, microbe we have identified against 11 antibiotics that perhaps are commonly used in India. And you could say, okay, none of them do anything, so then please don't prescribe them. Or this one is very effective, that's the one to prescribe. This is a slide indicating why we are using this combination of chemical plus mechanical, which we call universal lysis. See, gram-positive bacteria have thick cell walls that require mechanical plus chemical. For gram-negative, you don't need to do that. You can get away with just the mechanical. So the moving metal disc and silicon zirconia beads cause shear forces. So instead of here, the glass beads I showed you earlier, we use these silicon zirconia beads that cause the puncturing of the cells. Here you can see that the ribosomal complex is coming out, and then the sodium hydroxide releases the RNA fragments. Then we neutralize it. That prevents the hydrolysis of the RNA, and then we are ready for the detection. Good. I will skip a lot of stuff here because of the time. This I will briefly talk about. We've also been able to speed up the incubation process. So instead of putting a traditional incubator, we have the CD put in a special reader, and we put a membrane on top and the bottom that's gas permeable for aeration of the bacteria. And so we make sure that we learn the exact amount of nutrient for them. We prevent the formation of biofilms and clumping, and we do good temperature and environmental control. Doing that, we have learned that we can actually decrease the incubation time by quite a bit, which is further, indication that this is really a very good platform. This movie here shows the CD spinning in an incubator, a prototype, and it shows the disc oscillation for strong mixing. So strong mixing during incubation is also very, very important. So we have nearly a 2 times improved growth in 90 minutes compared to traditional shaker spin stands. And we have a reduction in assay time by 24 minutes. I'm going to skip all of that. This shows the system. This shows the workflow. And again, this was all built by our students in-house. And I'm kind of finishing off with some pictures. This is my team. Especially like to thank here Horacio Kido, who's always been our man to go to for system integration, and Sasha, who was a major driving force, who works now at a new company that we co-founded and is funded by Larry Ellison from Oracle, helping on developing CDs for COVID. This is my collaborator and good friend Satad Al-Saha. Perhaps some of you know this lady. And this is standing in front of one of the health kiosks in India. This is, so many friends, I would have to go over each of them. But let me just single out Amitabh Ghosh right here, who introduced me so many years ago to India, and had a long collaboration ever since. So with that, I'd like to thank you. I'm gonna stop sharing and I can take questions now. Oh, hey, Suma, how are you?
Yeah, hi, Marc. How are you doing? So
Good.
nice to see you. Nowadays, only online presence, but still very happy to see you here. And
yeah, same here. We have actually been teaching.
So are you teaching physically or?
Yeah, physically. Yeah.
Okay.
Actually, I have a class at 2 o'clock this afternoon.
Okay. Very nice. And this year we have missed you in terms of your presence in India.
Yeah, I know.
but,
Amitabh absolutely wants me as soon as possible. So,
yeah, but perhaps next year would be better for you to travel.
yeah.
I mean, we have your position active at IIT Kharagpur. So at
Thank
any
you.
point of time when you are, you are having opportunity or sort of, I would say, the courage to travel, such a long distance, you let us know, and we will be very happy to host you here. And just for the sake of all the participants in this course, of course, Professor Marc Madou is a name which all of you are familiar with, and there is no doubt that he is the pioneer of this lab on a CD technology. And Marc, this is a sort of faculty induction type of program, or you can say a short course, very much focused for those who are new entrants to this subject. So this is about the lab on a chip technology, this particular short course. And for the interest of all of you, I myself learned my first practical lessons on lab on a CD, and lab on a chip in general, and lab on a CD to be specific, from Professor Marc Madou only. So irrespective of our very friendly relationship, he remains to be like my teacher, and I accept Professor Madou to be one of the great teachers whom I have ever come across. I think we first met in 2004 in one of the Indo-US meetings organized at IIT Kanpur by Professor Ashutosh Sharma. And from then onwards, it has been a great learning. And I believe that this particular audience has greatly benefited from what Professor Madou has discussed. Now, Marc, I leave it on you. You ask 3 to 4 questions, and we will mark those participants who have answered those more or less correctly, and we will give them special recognition in their certification. So some simple questions from what you have covered so far today.
Okay.
Maybe something, some simple ones.
Okay. Let me start with perhaps why is the issue of bubble formation less of a problem when we use this specific type of platform? Because in all microfluidics, one of the big issues is bubble formation. You pipette in something, and almost always you get some bubbles to go with your sample. But here, this is very readily taken care of. So that's the first question. Should we wait for answers? Or
yeah, so some of you, please raise your hand, or you can simply unmute and try to give this question on bubble formation. A very, very practical question on these technologies. So you try to put forward an answer, please. Any one of you. Even the student participants from IIT Kharagpur, I can see a few of them. So any one of you. Professor Madou will not mind if you are if you are wrong, or
yeah, come on.
Yeah, I can see Preeti and Sujata answer.
Yes. You can unmute yourself and maybe switch on your video so that Professor Madou can see who is answering, and then you give your answer.
Good morning, Professor.
Morning.
The session was quite interesting. I belong to pharmacy background. I'm not sure, but I assume that bubbles will not form because of uniform mixing and the small space available in the CD. Whatever space is available, and the CD, whatever force we are applying, the centripetal and centrifugal forces, that mixing is proper, which will avoid bubble formation.
I like to hear another answer. Someone else?
Yeah, anybody else who wants to answer this? Otherwise, Professor Madou will give a very short explanation before moving to the next question.
I think Pinky has unmuted herself. She wants to answer or not?
Yeah, I think I agree with what the earlier participant said. Maybe even if it is there, practically when you're doing the experiment, maybe you can run it at a low RPM to to move the bubble away from the inlet side to the output side, maybe.
Right. I'll give
If
you a
I'm-
hint. Think a little bit more. What happens in centrifugation with heavier and lighter particles? Right. Now I'm giving you a really strong hint. Right. So if you imagine liquid volume and an air volume, right, and you have a
Yeah.
gravitational force, right, by spinning, what will happen?
Yeah,
You got it right.
the inertia is going to continue.
Yeah.
I mean, it will move away from the radius. I mean, from the center towards the periphery of the disk. It will move out, even if it is there.
Right, but what direction?
From
If you do centrifugation, where do the heavier particles go?
Center to the, it's away from the center rather, outward direction.
The liquid is heavier than the air bubble, right? So the liquid will go outwards and the bubble will go to the center, right? It will vent out.
Yeah. Correct, yeah.
Very good. Okay, so I'll be ready for a next question. So, you know, I was pointing out when you spin that disk, obviously the liquid wants to go to the rim, right? That's the centripetal force. You know, if you're sitting on a joyride, the force tries to, you know, remove you from the platform, so to speak. But I was showing you this trick that I want liquid to go back against the centripetal force, right? Could you give me a couple of ways on how you could do that? So I gave you one in the talk, and you could use that as an example on how it works, but there's other ways, right? So I have that platform, liquid goes outside, but I'm not finished. I want it to go back. What could we do? Use your imagination here, because there's several ways that one can do it. The one I illustrated is perhaps maybe the simplest one.
You can change the angle.
Say it again.
Change the angle.
Same.
If I'm doing it at horizontal, I change it to another axis.
No.
No.
That will not bring the liquid back. There will be still the centripetal force pushing it out.
It will be still outward, yes.
Yeah.
Yes. Yeah.
So yeah, the inward pumping with the centripetal platform. Make some drawings, use your imagination. It's kind of a very good exercise to start appreciating what all you can do with this platform. Mohan? I think Mohan Sumant is connecting, right?
Will deacceleration can I deaccelerate the disk and expect things to?
No, it could be teeny little bit, right? Because in acceleration, deceleration, you use the Euler force and you could spill over something kind of. but you won't be able to go very far back to the center.
Can we degas in a reverse direction?
Degas in the reverse direction. In other words, you would pull a vacuum on it, right, to pull it back.
Yeah, I think.
Right, but that's complicated, right? But yeah, okay. So at least we are thinking, right? We need an opposite force, right, to push it back. And so what you said is, I'll suck enough on it to offset the centripetal force, right? Any other answers?
Do you do it during the spinning of the disc itself? It happens during the spinning itself?
Yeah.
Or you?
Yeah,
Okay.
because otherwise, well, I mean, I grant you, if you have a technique to bring it back, I let you to, okay, you can stop it. I'm making it easier.
Can we use some sort of electric field, external electric field? Can I use that?
Yeah, see, there we go. We're starting to think. But it will be hard to do because typically for electroosmosis to work, your channels need to be quite small, and then in a way you're losing all of the advantages of the CD platform because electroosmosis depends on the type of liquid in the channel. But yeah, I'm giving you a point for that.
Thank
There's
you.
more. There's more.
We spin it and we stop it, and the force will die down. The liquid will come back to original, maybe not original, but it will move back then because it's not experiencing any force now.
Mm-hmm.
So it will.
But how do you make it move back?
Come
right?
back optical.
Yeah.
You again spin it. You spin stop, maybe, I don't know.
No, but then you will repeat the same thing, right?
So
And it will not go.
Another. I can guess another point, that is the centrifugal force that is proportional to either mass or the velocity. So can we reduce some mass flow in some sense? If we are not deaccelerating, can we reduce in its mass flow or something?
I'm not sure what you're driving at. Why don't I share again and kind of, I see there's also someone in chat here, reducing the RPM, Monica says. Let me go back and share my screen again and kind of point you at the right slide to think about. Here. You know, what we're talking about is, so normally liquid goes like this, right? Pushing it out. But I want to bring it back up. And you can see I've done that here. See? I brought it back up. So did you understand why it goes back up? So let's do it together, right? I'm loading, liquid goes down. And here there's some air, right? This air is trapped. Push it down, go even faster. And this liquid pushes against the air, right? Go even faster, I compress it even more. So what do I need to do? Go from D to E.
So air is try to expand again.
That's and compress. Yeah.
So what do I need to do to go from D to E?
Slow down.
Yes, there we go. Slow down, right? Because then the centripetal force is lesser. And the expansion here, this is just, you know, the gas law, you know, this can expand against the centripetal force and push back up. Good. So that was the second question. So we got that one. Now for the third question, I mentioned, you know, a lot of advantages of the CD platform. Could you imagine some negatives, some problems with it? What do you think might make it actually not all that easy to work with?
Is it single time use? Single time?
Right, but that is in a way pretty much because it's been in contact with blood and people do not want to reuse these things. But yeah, other things?
Is temperature affect the fluidity of this liquid?
I didn't hear it. I did not understand. Can you say it again?
Is temperature affect this fluidity of this liquid that we use?
We try to, yeah, but very little. But yeah, I accept that because, you know, it's based on viscosity and so, right? And anything that changes the viscosity or density of the liquid would have an influence. Yeah, good. More? Other ones?
Is disposal a problem?
Not really. You actually, you know, it's like lateral flow assays. You will never use these things again. In clinical settings, you often do not want to reuse your tests. That's not really a problem.
Yeah.
Other ones? I'll give you a hint. Suppose one of you was suggesting using electricity on the platform. So how could I bring electricity on the platform? Huh? It's difficult, right? Because it's spinning. You have to come up with some clever tricks on how to accommodate that. Okay, I think that's good. Thank you so much, guys. And hope you enjoyed it.
Thank you so much, Marc. And no, it was simply great. And all the best for the remaining, part of the year. And hopefully, hopefully in the coming year. Why I'm saying is because I don't see any physical sort of any possibility that we meet this year. But
that's right. Yeah.
very unlikely. But next year with a new year, new vision and a new hope, let us make a plan so that we host you at Kharagpur for a reasonable period of time. And our students and me personally, all of us are very, very eager and looking forward to you. So
very
have
good.
A nice
thank
day
you so much,
and,
Suman, for all of the nice words. And we had a good group, a good attendance here. That's always comforting. Thank you so much. Bye-bye.
Excuse me, sir.
Goodbye. Goodbye.
Thank you, sir.
Excuse me. Sir, may I ask a question?
Hello.
Hello. May I ask a question?
Yes, please go ahead.
Sir, which kind of detectors we have used in this CD technology?
Oh, we use fluorescence.
Fluorescence.
Yeah.
Okay. Thank you.
Thank you. Bye-bye.